Thank you so much, Julie, for introduction. Yeah, I wish I could present my findings in the San Francisco and meet with everyone in CFRI community. So I was really looking forward to doing that. But yeah, but let's start my presentation. So yeah. Today's my presentation focused on two topics. Mainly the number one, what cell type express CFTR in human airways. So for introduction, let me explain the brief structure of human airways. As you can see, there is difference between in large airway and small airways and alveoli in human airways. So in the big, large airways, the cell height is higher than small airways, and then there are relatively many large granule containing goblet cells and then many ciliate cells. And then if you go down to deeper lung, there is a different shape of cells here. So there are still ciliated cells and then dome-shaped secretory cells, which is also called club cells. And then if you go down more distal areas, you can see more cuboidal cells. And then this area is directly connected to the alveoli. And then one of our interest gene is SCGB1A1, which is also known as CCSP. This is club cell secretory protein. This is a marker of secretory cells. So we've been focusing on these cell types, and then that study was also supported by CFRI in previous 2 years. And then where is the threshold, like cut off of human large and small airway is 2 millimeter. And then the bigger than 2 millimeter airways has cartilage and the submucosal glands. And then more smaller airways than 2 millimeter is classically defined as small airways. And then the number of the one feature of small airway is even though the diameter is really small, but the airway is divided 23 times to the alveolar. So the number and the surface area of small airway is actually exponentially higher than large airways. Although this image shows the most of area is dominated by proximal airways, but the total surface area is much higher in this only like 2 centimeters from the distal region of the airways. So that's a unique characteristics of the lung. So and then the important innate defense mechanism of lung is mucociliary clearance, which is composed of 3 major cellular activities. One is a mucin secretion into mucus layer. So the secretory cells or goblet cells produce mucin, and then they secrete those mucin into the mucus layer just above the airway epithelial cells. And this can trap the pathogen or the chemical pollutant to remove those pathological stuff to the oral direction and then get rid of from the body. And then the second cellular activity is coordinated ciliary activity. So these mucus was actually carried by this cilia by moving really coordinately well. And this is also important component. And then the last one is a transepithelial ion and fluid secretion or absorption mechanisms. This fluid secretion is mainly regulated by CFTR. So those 3 components interact with each other and then maintain the airway mucus homeostasis to get rid of the irritant particles. And this, especially this fluid secretion regulated by CFTR important in CF pathogenesis because if CFTR doesn't work well, but still mucus is produced by some like virus or bacterial infection or other reason. So the mucus has been accumulated, but not hydrated by fluid secretion. Then mucus it's getting thicker and then eventually compress the cilia region, which is called periciliary layer. And then this cilia cannot get rid of the mucus above the cilia, and then this mucociliary clearance actually stop, and then repeated bacterial infection happen here. So this CFTR function is critically important to maintain this airway homeostasis. And actually this in vitro data shows this green region is a periciliary layer, like this region. In the CF airway epithelial cell shows depleted periciliary layer. So the cilia is totally compressed in only this thinner layer, and then these cilia cannot move the mucus anymore. So there is no doubt about the importance of CFTR. However, although the many technology is improved, but still there is a controversy surrounding the CFTR expressing cell type. So if we just look at the history of CFTR expressing cell type candidate cells, in 1990s, some reports by Dr. John Rengerhardt shows CFTR is expressed in non-ciliate cell in bronchiole, which is a small airways, and also he found a really high intense CFTR positive cell in submucosal gland ducts. But after 2000, there is another set of papers showing CFTR is clearly expressed in these ciliated cells and then not in these MAP5AC positive goblet cells. And also interestingly, this paper also detected this really high intense CFTR positive cell in the apical location of large airway superficial epithelium. And then they call this cell type as hot cells. And then just 2 years ago, using the single cell RNA sequence technology, some groups identified a new cell type, which is called ionocytes, which express really high CFTR expression compared to other cell types. So over time, there's been change in candidate cell type for CFTR expression, but still which cell type is predominantly express CFTR in all airway epithelium and then predominantly function. So those still remain unknown. So then we focus on this CFTR expressing cell type in normal human lung tissues. So and also the Ionocyte paper shows interesting functional data. So they modify the proportion of ciliate cell number using DAPT reagent, and then if you decrease the ciliate number, the CFTR function also goes down in this DAPT experiment. And then this is correlated with the number of ionocyte, but not correlated with ciliate number. So that means ciliate cell and CFTR function is not correlated. So those are already not consistent with historical data. So, but this data shows only ionocyte or ciliate cell numbers, but actually there are a lot of other cell type in these cell cultures. So still there are no definitional data yet. So we use multiple methods to identify CFTR expressing cells in normal human lung airways. But firstly, we also use single cell RNA sequence analysis, including drop sequence and 10x genomics. So briefly look at the method of single cell RNA sequence. So there are a lot of different cell types in human tissues. So firstly, we dissociate cells to single cells. And then if we run single cell in this mixed system with beads and oil to make one oil that contains one single cell and one barcode bead. This bead has a unique barcode. And then if we collect all these droplets, if you look at the one droplets, so this cell is immediately lysated by enzyme. And then the all RNA molecules are released from this cell. And then those molecules bind it to this barcode molecule, like barcode nucleotide. And then each molecule is barcoded to each cell, and also each molecule has different sequence. So after this, we lysate all droplet and then get everything together, but still all molecule has unique barcode. And then we can track each molecule is coming from each cell, and each single cell. So that way we can get the RNA gene profiles per each cell, single cell. So that's how single cell RNA sequence works. And then what we did first is we collected, we are interested in the difference between large and small airways. So we collected large airways and small airways separately. The large airways were collected from healthy volunteer by bronchoscopy. And then small airways were obtained from the lung transplant donor lungs by microdissection. And then this is first data of single cell RNA sequence combined with large and small airway samples. And then if we annotate each cell cluster, each dot shows each single cell. And then each cell that has similar gene profiles, over 5,000 genes located across each other. So we can annotate each cluster based on the canonical cell type markers. And then as a result, this big cluster is ciliated cells, and then roughly this portion is basal cell cluster, and then this portion is secretory cells. These three major clusters are already known major cell types in human airways. And also we have ionocyte, which has high CFTR expression, and also cycling cell types. This is kind of transitional cell type between ciliated basal or secretory cells. And we also have undefined cell type, which has higher mitochondrial genes. And in our study, so we haven't identified this cell type and then just call as undefined cell type. And then the proportion of each cell type is shown in this pie chart. You can appreciate more than 50% of cells are assigned to ciliated cells. That's really consistent with in vivo morphometric data. So that means we could collect all the cell as similar cell type as possible to in vivo situation. Also, small airways, the cell proportion, it looks similar, a little bit more basal cells in small airways than large, and then less ciliated cells compared to large airways. These cell types are assigned based on these canonical cell type markers, as I said. And then the unique data in our data set is we have large and small array data set. And then in order to confirm our small array is distinguishable from large arrays, and we look at the small array specific genes like SFTPB or SGB3A2. And then interestingly, there is no expression in these genes in large array data set, but there is a lot of expression only in small array data set. That is consistent with in vivo human lung tissue stained by RNA in situ hybridization. So this means our small array is small arrays. That's different from large arrays. But still, the major cell type composed of both large and small arrays, not so different in terms of major cell clustering, but there are different gene profiles as well in each cell type between large and small arrays. So, and then we plot CFTR positive cells on this UMAP clustering. So these fewer dots compared to this total cell is all CFTR positive cells. So you can see these cluster has more dots compared to these cluster or these cluster. That means these cells, these clusters have more CFTR cells. And actually, these pie charts show the proportion of each cell type for among CFTR positive cells. In large arrays, about 50% of CFTR positive cells are secretory cells. And then these less 30% of basal cells, especially these suprabasal cells that has a gene profile transitional between basal to secretory cell has dominated these basal cells which express CFTR. And in small arrays, even more than 50, about 70% of CFTR expressing cells are actually secretory cells. And then ionocyte express CFTR, but it's really rare compared to other cell types. And then in the ciliated cells, although the number of ciliated cells are a lot, but the proportion of ciliated cells for CFTR expression is much fewer than other cell type. And then these data shows expression level. So the previous data shows frequency of CFTR positive cells. We look at the intensity of CFTR expression in each cell type. So as reported by Nature Ionocyte paper, so ionocyte cluster express the highest CFTR expression compared to others, but rare, and these secretory cell has more dots, that means more CFTR positive cells, but the expression level is lower than ionocyte cluster. And then if we compare CFTR expression levels between large and small arrays, there is no statistical difference between large and small. That means although cell type might be different, but the total CFTR expression levels between large and small arrays are almost same. And then we combine the information of frequency and the expression intensity for CFTR expression, and then summarize the data. So this data shows in large arrays about 40% of CFTR contribution is originate from secretory cells, and then next contributor is basal cells. And the ionocyte express the highest intensity, but it's rare. The total contribution is around 8%. And in small arrays, interestingly, the contribution of secretory cell get bigger than large arrays. And then the rest of the proportion is similar order, like basal cell, secretory ciliate cell, and then ionocyte. So these frequency and intensity data suggest secretory cell is the major cell type for CFTR expression, RNA expression in both large and small arrays. And then the secretory cell contains mucin-producing cells. So we specifically looked at the mucin expression, the CFTR expression. And these RNA in situ hybridization images show CFTR is localized with MAC5B and SGB1A1 positive cells in large airways. This is consistent with single cell RNA sequence data. And in smaller airways also, this SGB1A1 positive secretory cells also express CFTR. And if we specifically look at MAC5B, MAC5AC, these are two major secretory mucins in airways. In large, these are total cells. About 30% of cells are expressing either 5B or 5AC in both large and small airways. And then these pie chart shows proportion of 5B or 5AC positive cells among CFTR positive cells in large and small airways. In large airways, about 35% of CFTR positive cells are also expressing 5B or 5AC. And in small airways, this percent is increased about to 70% of CFTR positive cells are expressing either 5B or 5AC. So these data suggest, at least in terms of expression, secretory cells which express secretory mucins also express CFTR. And interestingly, we have ionocyte cluster, but ionocyte cluster shows not only ionocyte genes, but some neuroendocrine cell genes. And then if we look at the difference between large airway ionocyte cluster versus small airway ionocyte cluster. So this heat map shows the difference between airway region and then ionocyte or neuroendocrine cell marker genes. So this heat map shows this region. Neuroendocrine cell markers are higher only in small airway cells. And then this volcano plot also consistent with almost all the CF ionocyte markers are highly expressed in large airway cells in ionocyte cluster. But neuroendocrine cell marker is highly expressed in small airway ionocyte cluster. So this suggests even in the same cluster of the same ionocyte cluster, but large area ionocyte cluster has more ionocytes, and then small area ionocyte cluster has more neuroendocrine cells. So there is a region-specific characteristics in terms of ionocyte and neuroendocrine cell gene. And then we looked at the smaller airways focusing on neuroendocrine cell marker protein. So consistent with this data, about 80% of small airway has at least neuroendocrine cell marker positive cell clusters. This is called neuroendocrine body. So this is consistent with the data that shows small airway cell has more neuroendocrine cell genes. So, and then we also look at the human lung histology of the non-CF lung and CF lungs to look at CFTR expression, the FOXI1 expression, if this distribution is consistent with single cell RNA sequence data. So interestingly, CFTR is highly expressed in some of submucosal gland duct cells, which is consistent with Dr. Engelhardt's data. And then in the superficial epithelium, the airways, CFTR expression is scattered and randomly distributed. And then this expression appears to be similar to all the airway regions. But FOXI1 positive cell, that is also, that means ionocyte. Ionocyte is expressed in submucosal gland duct, which appears to be consistent with CFTR high cells. And then this number actually is going down to the distal lung. if we compare the quantification data of these large airway versus small airways for CFTR, there is no difference, but FOXI1 positive cells are significantly decreased in small airways. This is a non-CF airway data. So this bar graph shows CFTR expression constant to the all airway regions, but ionocyte number is decreasing to the distal airways. A similar trend was seen in CF lungs. And we confirmed these findings using immunohistochemistry. And then immunohistochemistry shows these apical CFTR protein expression in non-CF airway cells. But in CF cells, this CFTR protein, which is supposed to be expressed in apical membrane, is accumulated in the cytoplasmic region, which is consistent with previous report, the abnormal CFTR protein accumulation in the cytoplasmic region in CF cells. And in smaller airways, there is a slightly weaker staining, but still specifically higher compared to other cell type and the isotype control. So this shows dome shape cell express CFTR protein on the apical membrane. So which we think consistent with secretory club cell apical membrane. So and then we we try to confirm these data using a replication cohort of other single cell RNA sequence data from non-CF large and small airway from much the same donor lungs, collaborated with Dr. Strip in Cedars-Sinai Medical School. So we again get the large and small airway cells and then did the same analysis. And then this is a final data that shows a contribution of each cell type to total CFTR expression. In large airways, secretly is number one, and then the second is basal cells. These two cell type accounted for more than 50% of total CFTR expression contribution. But ionocyte contribution is bigger than our data set, but still less than secretary or basal cells. And then this ionocyte contribution to CFTR expression dramatically decreased in small airway dataset. And then in contrast, secretory cell contribution was increased in small airway. That's also consistent with our single cell RNA sequence and the morphometric data. But here, so there is a limitation in single cell RNA sequence study. So single cell RNA sequence study cannot capture all the molecule in each single cell. And especially that is highlighted to when you try to detect low abundant genes such as CFTR. So this is our single cell RNA sequence data, and then this is a Cedars-Sinai single cell RNA sequence data using we use a drop sequence, and then they use a 10x genomics platform. There's two different platforms, but we identified 950 cells among 26,000 cells. That is 3.6% of total airway epithelial cells express CFTR. And in their data set, 10x genomics is a little bit better capture ratio and better depth to detect the low abundant genes compared to drop sequence, but still their data shows 6% of total cell express CFTR. So, but as I mentioned, the single cell RNA sequence cannot detect all the low abundant genes. So we suspect there might be more cells expressing CFTR in vivo situation, because CFTR is important for mucociliary clearance, and only 4 or 6% of cells expressing CFTR, that might not be enough to maintain all the mucociliary clearance by fluid secretion. And then we came up with site spin plus single RNA inside hybridization experiment. So we dissociate cells like when we use for single cell RNA sequence, and then fix the cells and then perform RNA inside hybridization, focusing on CFTR and cell type markers. Interestingly, this experiment worked pretty well, and then we can identify CFTR messenger RNA signals in each single cells, and also cell type marker consistently show. For example, this is FOXJ1, which is a serrated cell marker, is highly identified in serrated cells. So now we use this method to systematically quantify which cell type express CFTR. And then we use 4 different cell type markers. One is secretory cell type marker, SGGB1A1. This is a representative image. So, and then also basal cell marker, keratin5, and ionocyte marker, FOXI1, and serrated cell marker, FOXJ1. So these are distribution of number of dots per each marker positive cell. This the x-axis shows the number of dots, and the y-axis shows number of cells. So this distribution shows SGB1A1 positive secretory cells have more dots per cell in both large and small compared to other cell type. And then FOXJ1 positive cell has more like 0 positive cells. That means the FOXJ1 positive serrated cells tend not to be expressed in CFTR. And in contrast, this FOXI1 positive ionocyte express more dots per cell in especially large airways, and then small airways shows only 16 cells in all the slides we studied. These are summary data. This is frequency of CFTR positive cells among each marker positive cells. About 80% of CCSP SGB1 positive cells also express CFTR. In contrast, serrated cells, about 30% of cells express CFTR. We have to be careful. We use only one cell type marker and CFTR at the same time. So there could be the co-localization of 2 different cell type markers in each cell, like transitional cell. So we might have missed those transitional cells or overcounted those cells. And then this is the intensity of CFTR expression. The ionocyte express the highest number of dots per cell. And then next one is a secreted cell type. And these are higher than serrated cell or basal cells. These are also, this order is consistent with other data set. And then next, we also use a more sensitive method like single cell RNA inside hybridization in single cell based more sensitive method. This is a single cell QRT PCR. So we get the large airway single cells from healthy volunteer by bronchoscopy, and then sort single cell, and then just put each single cell into each well in the 96 well plates, and then run qPCR per each cell, and then look at the gene expression for our target genes. So we use this strategy. We studied 264 cells from 2 healthy volunteers, and then we did the quality control using a GAPDH and the EpiCam positive cells, and only 146 cells passed this quality control. And then we assigned each cell based on cell type markers, XION positive cell, and then negative for all other 3 cell types, or other SGB1 positive, and then all negative for other cell type, like that. And then we could not have any ionocytes that reflects the rarity of ionocytes, but we have 52 exclusive secretory cells and 39 exclusive basal cells, and then this is a small number, but 8 exclusive FOXJ1 positive cells. And the others contain the cells which express at least 2 cell type marker genes. And this is the frequency of CFTR positive cells in each cell type defined by this definition. That consistent with other data, secreted cell, about 80% of secreted cell express CFTR, and then 50% in basal cells. But among only 8 cells, but no cells express CFTR in serous cells. And then these transitional cell type also express CFTR. And this is the intensity of CFTR expression in each cell type. So this also consistent with the frequency. So, and then this, for your information, the GAPTH gene expression is similar, no different between groups. That means that these seed cells are still viable, and then the quality is not bad. And this major cell type marker is consistent with this cell type definition. So these two more sensitive methods than single cell and sequence agree with secretory cell express CFTR most dominantly. So the next we try to relate these expression data to functional data. So we have developed different cell culture systems for large airway and small airways. And then we compare the gene expression for these two culture systems. So these are small airway specific genes. Even in vitro, these genes are specifically higher in these small airways. But SGBY1 expression is similar in both regions. And then here CFTR expression is also same. This is consistent with single cell and sequence data. So there is no difference in CFTR expression between total large air cells and small air cells. And interestingly, these FOXI1 and SSCL3 is ionocyte markers. These genes are significantly lower in small airway. That's also same as single cell data. So despite the lower gene expression of small airway cells, but still CFTR expression is the same. And then if we look at the function, small airway shows the similar, like significantly similar CFTR function that match to large airway cells, despite the lower gene expression of ionocytes. That not directly, but that suggests non-ionocyte CFTR positive cell has a function for CFTR in small airways especially. And then next we utilize the already known nucleotide that can regulate CFTR activity mediated by specific receptors such as P2RY2 and adenosine receptors. So these are already known. The CFTR secretion is mediated by these receptors by nucleotide. So because why we focus on these receptors is these single cell and sequence data. So our single cell and sequence data shows ionocyte express the highest CFTR and then SLC12A2. This is a chloride ion entry channel, which is important for sustained CFTR activity. So these are high, but these P2RY2 or adenosine receptor is much lower than these secretory cells. In contrast, secretory cells express CFTR that's already confirmed by our other data sets, although this heat map shows very weak expression, but these cell type express these P2RY2 or adenosine receptors that is important for CFTR activity. So, and this data is consistent with other published single cell and sequence data. This is Dr. Sripsida's data. Also show ionocyte is almost undetectable expressed P2RY2 and acapulin 5. And this is another data set published by bioRxiv. Ionocyte express high CFTR but low P2RY ADORA2B. So, and then we looked at P2RY2 expression, for example, for each cell type using the single cell QRT-PCR, which was used previously, the secretory cell definitely express P2RY2. And also we look at each single cell for these receptor gene. So SCGB1 positive cell co-localized with these P2RY2 receptor genes, but ionocyte, FOXION positive ionocyte didn't express these genes. And also adenosine receptor is also highly expressing secretory cells, but not ionocyte. So there could be difference between these receptor expression by cell type that suggests CFTR might be differently regulated by cell type. But at least now we know secretory cells express these genes, receptor genes. We stimulate these genes with a nucleotide. So these oozing data shows if we block calcium activated channel because the nucleotide like ATP or UTP activate not only CFTR but calcium activated channel. So we firstly block calcium activated chloride channel using adenine and then add UTP, and we could see CFTR response that can be inhibited by CFTR inhibitor. And then these response was not seen in CF cells. So,
Kenichi, can I interrupt for a moment?
Yes.
I just want to let you know that we have another talk starting,
okay,
Dr. Conrad. So yeah, if you could just, you know, wrap up as.
Yes, sorry.
That's all right. Yeah. Just wanted to let you know.
Yes. These data suggest the nucleotide can activate CFTR, and then our previous this receptor data combined with this receptor data. So secretory cell is a major cell type. This where this nucleotide regulated CFTR response happened. So and then the conclusion is combined with these functional and expression data, CFTR is expressed in multiple cell type in human conducting airways, but particularly in secretory cells. And the secretory cells dominate airway CFTR expression and the function in both large and small airways. So yeah, let me skip these slides. So yes, I'd like to appreciate all the collaborators and mentors, Dr. Bush and Wanda O'Neill, and all other collaborators inside UNC and outside UNC. And of course, I would like to thank you, CFRI, for giving me such a great opportunity to study this CFTR mechanism study, and also give me a chance to present our findings here. Thank you so much.